Okay, dear students, in the previous class we discussed about the structure of DNA molecule, and in this class let us study about packaging of DNA helix. Packaging of such a large quantity of DNA, it is packed in the chromosome, inside the chromosome. There is a systematic pattern for packaging. Let us study how it is packed inside the chromosome. So, the structure of packed DNA will be like this. So this is DNA and this is H1 histone protein, H1 histone. This is histone octamer. This is core of histone octamer. Core of histone octamer. And this entire structure is called nucleosome. The entire structure is called nucleosome. In prokaryotes, prokaryotes example is, E. coli. In E. coli, DNA is not scattered throughout the cell. DNA is not scattered. Is not scattered or spread. DNA being negatively charged is held with some positively charged proteins. They are called nucleoids. Positively charged proteins, and that positively charged proteins and DNA together it forms nucleoid. The negatively charged DNA combined with positively charged proteins called nucleoid, positively charged proteins to form a structure called nucleoid, a structure called nucleoid. So, the typical, on the other hand, in eukaryotes, as there is a set of positively charged proteins. Positively charged proteins called histones. Which pack or which is associated with negatively charged DNA, which is associated with negatively charged DNA. Yeah, negatively charged DNA. And always histones are rich in basic amino acids called lysins and arginines. So histones are rich in basic amino acids. They are called lysins and arginines. So here we should remember, in prokaryotes what happens? DNA will combine with positively charged proteins, and the DNA along with the positively charged protein, it forms a structure called nucleoid. So that you should remember. It forms a structure called nucleoid. On the other hand, in eukaryotes, there is a set of positively charged proteins. They are called histones, which is associated with negatively charged DNA. DNA is negatively charged in both prokaryotes as well as eukaryotes, but the associated structure along with the DNA in prokaryote is different from eukaryote. In eukaryote, what is there? Histone proteins are there. Okay, histones are how they are prepared. They are rich with basic amino acids. Those basic amino acids are called lysins and arginines. Now, they may ask a question. Define nucleosome. So they may ask a question, define nucleosome. If that is a question, then your answer must be like this. What is the question? In the exam, they may ask this for 1 mark. Define nucleosome. Negatively charged DNA is associated with positively charged histone protein to form a structure called nucleosome. Negatively charged DNA is associated with positively charged histone proteins to form a structure called nucleosome, where the nucleosome is made up of amino acids, especially the rich amino acids are lysines and arginines. So if you write this much, one nucleosome consists one nucleosome consists 200 base pairs. 200 base pairs. One nucleosome consists of nearly 200 base pairs. Then the total number of nucleosomes in human body. Total number of nucleosomes in human body is equal to 6.6 into 10 to the power 9 base pairs by 200, that's equal to 3.3 into 10 to the power 7. 3.3 into 10 to the power 7. Now, what are the other aspects with regard to nucleosome that we should remember? So what is nucleosome? The negatively charged DNA is wrapped with the help of positively charged histone proteins, and it forms a structure that is called nucleosome. And one nucleosome consists how many base pairs? 200 base pairs. You know, base pair means adenine, guanine, thymine, cytosine. Adenine pairs with thymine, guanine pairs with cytosine. Adenine pairs with thymine with 2 hydrogen bond, and guanine pairs with cytosine with 3 hydrogen bond. So next, nucleosome contains the repeating unit to form chromatin. Chromatin is thread-like stained body. Nucleosomes contain repeating unit to constitute chromatin, constitute to form chromatin. Chroma means colored, chromatin means colored body. Chroma means colored, chromatin means colored body. What is chromatin? Chromatin is thread-like stained or colored body. Stained or colored body. This is very important. Chromatin is a thread-like stained or colored body. Then nucleosomes in chromatin appears like beads in a string. Nucleosomes in chromatin appears like beads in a string. Beads in a string. Beads in a string. So nucleosomes in a chromatin appears like beads in a string. String means a thread. Beads means the round ball like structures. For example, ಈ ಸರದಲ್ಲಿ ಇರ್ತದಲ್ಲ ಮಣಿಗಳು, ಆ ಥರ ಮಣಿಯ ಸರದ ಹಾಗೆ ಕಾಣಿಸ್ತದೆ. Nucleosome in chromatin appears like beads in a string. Okay. Then chromatin is packaged into chromatin fibers, coiled and condensed at metaphase stage to form the chromosome. So how this chromatin is packaged? Chromatin packaged into chromatin fibers, these chromatin fibers coiled and condensed, coiled and condensed in metaphase stage. In metaphase stage to form the chromosomes. To form the chromosomes. What is condensed? Large quantity of the object is accumulated in a small area, and that object will become dark. So chromosomes appears to be dark in microscope in metaphase stage because large quantity of chromatin fibers were accumulated in a small area. That's why it appears to be dark. So chromatin is packaged in such a way that the chromatin fibers, they accumulate at one particular area and or inside the chromosome or one particular area to form the condensation of the fibers, and those condensed fibers will be clearly seen in the metaphase stage, and it forms chromosomes. It forms the chromosomes. Now let us study what happens next. High level of packaging of chromatin requires non-histone chromosomal protein. High level packing of chromosomes, high level of packing of chromosomes, that means more complicated level of packing. Normal level is with chromatin fiber and histone protein, but still more further complicated level is associated with packing of chromosomes is associated with NHC. What is NHC? Non-histone proteins is associated with non-histone proteins. Is associated with non-histone proteins. Then this is very important. The high level of packing of these chromatin chromosomes are associated with NHC proteins. What is NHC? It is non-histone proteins. Then there are two regions in chromosomes. Chromosomes include two areas. One is euchromatin. Euchromatin. They may ask define euchromatin, or they may ask write the differences between euchromatin and heterochromatin. So euchromatin is loosely packed and transcriptionally active chromatin, lightly stained and transcriptionally active region. What is this transcription? It is process of production of mRNA from DNA. What is transcription? It's process of production of mRNA. What is mRNA? Messenger RNA. Messenger RNA is produced from DNA. So, what is transcription? It's a process of production of mRNA from DNA is called transcription. So, what is euchromatin? It is loosely packed and lightly stained and transcriptionally active region. It is called euchromatin. Then what is heterochromatin? Heterochromatin. It is densely packed and dark stained, densely packed, dark stained, inactive region of chromosome. Inactive region of chromosome. Inactive region of chromosome. So, densely packed, dark stained, inactive region of chromosome is called heterochromatin. So, there are two types of chromatins. One is called euchromatin and another one is heterochromatin. Euchromatin is loosely packed, lightly stained and transcriptionally active region. Heterochromatin is densely packed, dark stained, inactive region of chromosome. Inactive region of chromosome. So, this is with regard to the chromatins, euchromatin and heterochromatin. So, if they ask it for 3 marks, write the differences between euchromatin and heterochromatin. What should be your answer? Your answer should be like this. Under euchromatin, it is with loosely packed chromatin fibers. Under heterochromatin, it is with the densely packed chromatin fibers. That is the first difference. Second difference is under euchromatin, it is transcriptionally active. That means it is capable to produce mRNA. Under heterochromatin, it is transcriptionally inactive. It is transcriptionally inactive. And the third point is euchromatin is lightly stained. Under heterochromatin, you should write it is darkly stained. Under euchromatin, it is lightly stained. Under heterochromatin, it is darkly stained. So, there are 3 points. So, definitely, they may ask this question, write the differences between euchromatin and heterochromatin. This is a homework. You should write in your notes. Write the differences between euchromatin and heterochromatin. Write the differences between euchromatin and heterochromatin. If they ask this question, it can be of 3 marks. 3 marks. Then you should write the 3 differences which I mentioned. Is it okay? Yeah. Next, let us study about the search for genetic material. Earlier, before the invention of DNA, there was different thoughts with regard to the genetic material, and those thoughts they gave different ideas for the genetic material. Some people, they told proteins are the genetic material. Some people, they used to tell carbohydrates are the genetic material. But some scientists, they used to tell RNA is the genetic material. Some they used to tell DNA is the genetic material. But there was a lot of confusion. Nobody was able to conclude which was the genetic material. During that time, a scientist called Griffith, who accidentally conducted one experiment. Griffith's experiment. Griffith experiment. On the basis of transforming principle. Transforming principle. Griffith's experiment is based on transforming principle. So what are the experimental organisms he used? Griffiths used mice and Streptococcus pneumoniae. Streptococcus pneumoniae. Griffiths used mice, that is, and also the bacteria Streptococcus pneumoniae. In Streptococcus pneumoniae, he observed 2 varieties. Streptococcus pneumoniae. He observed 2 varieties. One is smooth strain or virulent. Smooth strain variety, or he called it as virulent. Virulent means which is harmful, which can cause death. So smooth strain or virulent, it has polysaccharide mucus coat around it. Polysaccharide mucus coat around it, which causes pneumonia. Second variety was rough or R strain. Rough, or here it is S, here it is R strain. Rough or R strain. It is non-virulent, not harmful. How it was? No mucus coat. No mucus coat was there. Mucus coat was not there and does not cause pneumonia. Does not cause pneumonia.